Okay, everyone, we're going to get started now with the next keynote speech to try to stay on time. We can all direct attention now and get started. That would be absolutely wonderful. Okay, thank you so much. So first of all, I hope that the day has been going well so far, that you enjoyed our first keynote speech from Dr. Varmus, and then also that you thoroughly enjoyed, hopefully, the speaker sessions. I found the topics to all be quite riveting and a quite nice variety. So it was really great to see all the different research that we have going on here at the symposium. And I'm also looking forward to seeing the poster board competition tomorrow. I think that'll be a spectacle. But with no further ado, I want to introduce our next very esteemed and distinguished speaker, Dr. Ruth Lehmann, who is a distinguished biologist and developmental geneticist who has made significant contributions to our understanding of germ cells, which I'm sure we'll hear a lot about today, given the title screen of this presentation. So she was the director of the Germ Institute of Biomolecular Medicine and the professor of New York University School of Medicine. But currently she's the director of the Whitehead Institute over at MIT. Lehmann's groundbreaking research has shed light on the molecular mechanisms that govern the formation and differentiation of germ cells. And it's her discoveries that have had very important implications on the future of reproductive medicine and overall fertility treatments. She is a recipient of many awards, including the Gruber Genetic Prize and the Wilczek Prize in Biomedical Science. And overall, Lehmann is recognized to be one of the leading experts in her field. And I don't know about you, but I'm very excited to hear what she has to say.
Thank you for that incredibly overly positive introduction. It's really great. It's also nice. I've just heard so many talks from you guys. It actually made me do something that you should never do, and that is I added some slides to my talk. We will see how well that goes. And the reason why you should never do that is because you may have made a mistake when you do something so late. But anyways, I thought I had prepared something which was very general, and then I thought, maybe some of you may be interested. So there will be a tiny little intro where I'm showing some data which are actually from my lab right now, what I'm excited about. And so, if you feel like, oh my God, I don't know what she's talking about, then just a few minutes later, I hope I can catch you back. So I'm going to tell a little bit about how I became a scientist and what I feel is important. But I wanted to really concentrate on the love of my life, which is germ cells. And so I want to tell you about that. And but I want to start off with my way. So now I'm going to ask a question. How many of you were not born in the US? Okay. So I was born in Germany, and I came to the US as an undergraduate, and that totally changed my life. I was a biologist, but I actually have to say I didn't even know what an experiment was. And it wasn't until I had a Fulbright fellowship to come to Washington University in Seattle that I found sort of interest in science and also realized what it means to make discoveries. And I think if you ask me why I'm a scientist, I think I'm a scientist because I like in the morning when I get up to know that I am driving why I'm getting up. And I'm driving why I'm getting up because I'm interested in a project. Nobody tells me what I have to work on. It's a little different when you have an administrative role, but when you're a scientist, you can do that. And that's what I really, really like. And I think it is that self-determination that I think, and the idea that you can make discoveries that really has driven me as a scientist. And it started really early on, actually, when I was in Seattle. So I was an undergrad. I had to go back to Germany, and I didn't know what to do now because I had to finish actually my undergraduate degree in Germany, and I really wanted to work in the lab. And so my advisor here in Seattle, he said, why don't you go to a conference? And at that conference, I met this woman, Christiane Nüsslein-Vollhardt, She was just starting a position at that time. And she couldn't actually take me as a graduate student, so I had to go somewhere else first. And then I finished my first degree, which is a master's in Germany. And then I did my PhD with her. And that completely, again, in a way, changed my life because I followed the passion that I had already, which is studying genetics. But now I was able to study how actually the body plan of the embryo is established. And so this is sort of one of the results that were part of my graduate thesis, and that had to do with how you make anterior-posterior polarity in the fly embryo. And so they have a head, and the larvae has a head and it has a tail, and we were looking for mutations which affected that. And so the first mutation that I found was this mutation, which the gene I later called Oscar, and here's the something literally. It's according to a novel from the Tin Drum from Günter Grass, and that novel and the reason, and so we all the fly genes, you probably know, the fly genes have funny names. Other the C. elegans genes have boring names, but the fly genes have these names which are. But then you have to know a lot about it too, which is sort of a problem sometimes. So in this case, you have to know why did she call this Oscar, in this book by Günter Grass, there is a character which is called Oscar, which can't grow. And that's why I called this gene Oscar, because as you can see, it lost all these abdominal things. So it's kind of short. And that was what we were looking for. So we were looking for how the patterning. But when I was looking at this more carefully, what I noticed, and it's very hard to see here in this image, there are these cells at the very posterior end of, this is a pretty early developing embryo, and they are called the germ cells, and those germ cells were also missing. And so from this analysis of pattern formation, all of a sudden I saw these cells, which are called germ cells, and I was wondering, maybe it would be really interesting to use Oscar and other genes in that group that we found to analyze how actually germ cells are made. And so that got me off, and sometimes people give you the advice that you should change your organism that you work on. I did not. I changed fields in terms of, I became a molecular biologist. I sort of just started to apply new technologies to whatever the problem is, whether it had to be structural biology, molecular biology. We do a lot of imaging now, which I will show you. So that's kind of how, I think Harold was talking about serendipity, and I think this is also a little serendipity. There's also something here about science, and that is sometimes it is really about observing something, and then you observe, and then you take that opportunity from what you observed. And so what I want to tell you is about germ cells, and I really want to sort of tell you something very specific, what we have found in the lab, but also in general want to talk about not just germ cells in the fly, which I'm studying, but sort of in general, what are germ cells? And obviously, when this picture of Adam and Eve, what that's describing is when sperm and egg gets together, it makes a new soma, and then it also makes the germline, and the soma will eventually die, and the germline will go on. And the soma also is usually the carrier of disease. The germline diseases are diseases of reproduction. So they're actually not affecting the organism itself, which is kind of very interesting to think about. It's really affecting the next generation. And so, first of all, a germ cell is not a germ. So often when I say to somebody, what are you working on? So I've stopped saying that. I don't say I'm working on germ cells. I say I'm working on the cells which make egg and sperm, because people go like, ugh. So it's not a germ. It has more to do with germinating, right? So germ cells are indeed the cells which make sperm and egg, and they are needed to then germinate an embryo, or in this case, a plant cell. And this happens when the egg meets the sperm, and the egg is much, much larger than the sperm, and I will come back to that later and tell you something more about, in principle, what the contributions are. And then what happens after that is obviously the nucleus of the sperm and the nucleus of the egg join, and then an embryo starts to develop and goes through many rapid divisions. And then eventually, so we have a ball of cells here, and then eventually cells become specified among that ball of cells. And some of the cells already in the early embryo get set aside to be the germ cells of the next generation. And we call those cells primordial germ cells. Because at that point, they're not egg or sperm. They're only on their way. And I tell you a little bit of how they get to that way to become egg and sperm. Because at that point, they really don't know even if they're supposed to be egg or sperm. And so this happens in every organism, whether it's a fly or it is a mouse or it is a human, and we actually know quite a bit about this now. And so I'm going to start off with telling you something about the specification, and then I'm telling you something about the life cycle of the germline, because it's actually kind of hard. You never know where to start, right? Should you start at the oocyte, or should you start at the embryo, or where should you start? Because it is a cycle. So and that has a lot of implications of how information is passed on, because this is the carrier of the information for the next generation. And so there have been many speculations, so here in long, long time ago, actually how it became clear that somehow sperm and egg had something to do with reproduction, but it was really unclear of what is it that carries the information. And so one hypothesis was this, which is called preformation, that there was already within the sperm a little homunculus was sitting there, a little human being was sitting there in the sperm. And somebody actually proposed, said they had observed this. This was when there were very primitive ways of looking at sperm. But if you think about it, that's kind of impossible that the whole organism sits there. And of course, now we know it's genetic information, which is sort of the homunculus, because it has all the information. But of course, the genetic information in the sperm is only half the information that's afterwards in the embryo, because there's also the information, the genetic information from the mother. So, sort of not quite a homunculus, but what then became apparent when people were studying various organisms, it became clear that there was actually something in the mother, so not in the sperm, but there was something in the mother, and that means in the oocyte, of many egg cells that was passed on to the next generation, which was absolutely important for making the germ cells. And that's called germ plasm or germ granules. And here you see it in the fly embryo, so that you see nicely at the posterior pole, which I showed you before, where those germ cells are. There's a region which is really intense. It has these granules. I'll tell you a little bit more in a moment about that. And that's where the germ cells form. But it wasn't restricted just to fruit flies. Nematodes and worms, it was the same. Here's these granules, a little different initially everywhere, and then more recently we learned that the granules disappear in the front part, and then they concentrate in the cell, which will eventually give rise to the germ cell. And here we have a frog embryo, and again you have this in the oocyte. We have the specialized region, and then the cells which inherit that region will become germ cells. And so that is kind of, I mean, it's not preformation in terms of like every cell already knows from the egg what it's going to be, but it is certainly, there's some information in the egg already, which in these cases has some activity which produces the germ cells. And so that here is sort of this diagrammatically during oogenesis, these factors are accumulating at the posterior end of the oocyte, and then in the embryo they form. And there is experimental evidence then to show that what is accumulating actually here is important for making these germ cells, and that is transplantation. So normally this part makes the germ cells, but now if you take this cytoplasm, this preformed cytoplasm, is specialized determinants for germ cell fate, put it at a new location, then germ cells will form there. Saying that there's something in the cytoplasm, so this is not genetics, has nothing to do with the DNA. If there's something in the cytoplasm which keeps some information which tells that region of the embryo to develop this germ cell fate. So that's one way. This is called either maternal determinants or preformation also. But then there's another way, and that was discovered when people started to look at how germ cells were formed in the mouse. And so in the mouse, there's an early stage here of development where this epiblast will develop the entire embryo. And then we have these extra embryonic tissues, which will, for example, develop all the placenta, the allantois. And what was found was that somehow there were signaling molecules in this extra embryonic space, and they were signaling to the cells in the epiblast. And it became clear that there was nothing in the egg of the mouse or the human which had this predetermined factors. And that's shown here. So these cells are somehow, they become either mesoderm or they become germ cells just because of the location, just because of the signaling. And so the similar experiment, so if you see something like that, you can say, oh, if I take that signal away, you don't get germ cells. That tells you the signal is necessary. But what is a real proof is to show that the signal is sufficient, like the transplantation experiment. So that was also done, and that is if you take cells from the bottom here and you put them actually into this region, then germ cells form there from these cells. So that clearly says that in the mouse, this is very different. So, and then it became clear in the human, it's very different. And so you could say, oh, maybe more primitive organisms have this preformation, and then you move towards this more inductive embryonic patterning system. But the surprise is that it is actually the inductive system which seems to be the primitive. And the arguments here, it's very always evolutionary because you can't really do evolution, right? But you can look at different organisms which separated at different years evolutionary. And so some experiments have now been done where it was asked whether you had this preformation pattern, so where the maternal germplasm was. And there's many different organisms, so it's not that vertebrates, I already showed you xenopus, fish has other vertebrates. But then for humans and mice, I told you, but then also very primitive organisms which were actually evolved way before the separation of the lineage which produced insects and vertebrates. They have also this inductive mechanism. So the idea is now that it is actually this inductive mechanism is the primitive way, and then this germplasm came later, and it's a little odd because why and what does this help? So some ideas are that this could be helping the embryo to develop maybe faster, or that there was some necessity at that stage to have actually more of a specification in those eggs than in the other eggs. But I will tell you at the end that it doesn't actually matter so much. So because irrespective of how the germ cells are formed, so irrespective of what the evolutionary path is, they all have to do the same thing, and there are also many genes which are involved which are exactly identical. So at the end, how the egg gives rise to the germ cells and how the embryo gives rise to the germ cell doesn't really matter so much. But there are these common factors. And what is really interesting is that sort of different from how we think about how all the cells in the embryo are developing, which is that there are specific transcriptional responses. That means genes are being turned on in the nucleus of a particular cell, and then that cell will commit to a cell fate. That's how the mesoderm, that's how our muscles are made, this is how our neuron cells are made. For the germ cells early on, it's very important to repress those signals, because when you think about a germ cell, it has to go through the whole cycle. So it has to be able to make all the cells in the body in the next generation. So it has to resist from that. And so it is transcriptional repression, which is one of the hallmarks of these early germ cells. But then what's conserved, so really at the molecular level, are RNA regulators. So it's post-transcriptional regulation, which kind of gets us again more towards the cytoplasm and away from the nucleus. We also, one other common part of multicellular organisms is the process of meiosis, where the paternal and the maternal genomes are mixed to make a new mixture of recombined progeny. And so that is actually a form of cell division, which is only found and only done by germ cells. Another aspect is, and that is an aspect which is really important when we think about the function of germ cells. So germ cells have to give on the genetic information to the next generation. And by doing that, there's two parts. One is the genome has to be really intact, right? There has to be quality control, because if the genome isn't intact, the organism couldn't develop. On the other hand, the germ cells are also the motor of evolution. So if we want to make any changes, it has to be through the germ cells, because that's how evolution for the next generation is driven. And so transposable elements play actually a very important role in the germline, and in pretty much all germ cells that have been studied, all the different organisms, it becomes clear that transposable elements, which are these elements which take up a lot of the genome, actually more than the coding sequences for proteins, they, on the one hand, can be a threat to the genome integrity. On the other hand, they can also lead to rapid evolution, and that's, at least that's what we think, is why they are particularly regulated in the germ cells. And finally, pathogen control is, of course, also an important aspect, because pathogens can be carried on, actually intercellular pathogens can be carried on through the germline. And then finally, there's the transgenerational aspect, of course. The genome is carried on, and the cellular machinery is carried on into the next generation. That's really important. And so irrespective of the organism, the primordial germ cells have to do these three things. They have to control the transposable elements, they have to repress somatic development, and then they have to keep the pluripotency of the organisms. And so now now comes this little segment where I'm going to talk a little bit about some work that's going on in our lab right now, and then we'll talk more about what happens to the germ cells after they're specified. And so one aspect which is really interesting, so I told you there's this germplasm, and you may say, what is the germplasm? Yeah. Of course, OSCAR plays a really important role in the germplasm of the fly, but germplasm, while in the early embryo, I told you for specification of the germline, it's not absolutely important, right? In some organisms, they don't have it. It's different evolutionary ways. But the formation of RNA granules, and this is the RNA regulation, is a common hallmark of all germ cells. So even the germ cells, like in the mouse or in humans, which form because they are induced by cell-cell contact, they have these granules. And these granules have components which are shared. So one is an ATP-dependent helicase called Fawzaan, which is shared between the fly germ cells and the human germ cells. And there's several other components. And it's very, very striking. And for a long time, it was more an observation where, in the mouse, for example, you see these, in the oocyte, you see these specific granules, which you also see in other oocytes. Then I showed you already these early germ granules, and then also during spermiogenesis, around the nuclei, irrespective of how the germ cells form, we find this granules which are called nuage, and their major role is transposable element control during spermiogenesis. And so what are these germ granules? So they are organelles, but they're organelles which don't have a membrane. And recently, it has become clear that there are quite a few of these membraneless granules in all cells. And they come together because of high concentration of proteins, which then undergo a liquid to solid transition, or liquid to gel transition, dependent on the conditions. And this is occurring in the germplasm of the fly, but it's also, for example, there are neuronal granules which have these, and then there are these granules that happen during stress in cells, the stress granules and the P bodies, which sequester RNA, or nuclear granules like the nucleolus, which is required for RNA synthesis. And so in all of these cases, they are membraneless, They come together by this condensation. They have in specific protein-protein interactions, but there is more to it than a stoichiometry of protein-protein interactions. So they are sort of molecular machines, but we don't understand them quite yet. And so there are quite a few of them, as you can see, and we're just starting to learn the grammar. We know now what kind of proteins are responsible for making these granules, but we don't know exactly how they're actually functioning. And so in all these cases, what we know is that the proteins which form these granules or seed these granules have RNA binding domains. So this is, again, this conserved RNA binding. And they also have protein-protein interactions. And then they have large unstructured domains. And so here is the fly. In the fly, we have this localized RNA in the germ granules. And you can actually see these granules here by EM. So they are kind of dense. And they have some ribosomes around them, and that suggested to us that actually this RNA, which is concentrated in the germplasm, would be translated. And you can see most of the RNA is actually everywhere. That RNA is not translated, only the RNA in the granule. And so we thought we had a unique chance for one of these granules to actually demonstrate what the function of it is, because that has been very unclear for most of these granules. And so our idea is then that you have RNAs concentrated in these granules, and that then, because of the granule formation, that the RNAs are translated. And so we used a method to observe at the same time where the RNA is and where the protein is translated, because the important question was, is the RNA actually translated in the granule? Because most of the other granules which have been studied, they do the opposite. They actually thought to repress or sequester RNAs. So we had to really prove that the RNA, and what we did here is a trick where we, at the same time, were probing the RNA with an antisense probe against the RNA, and then we put these sequences into the RNA, which are translating into a little peptide, and that peptide is recognized by a fluorescent tag. And so whenever the protein is made, as soon as the protein is made, the tag is recognized. And since we have 24 of these tags, that is a high concentration as long as the protein is made, because we also have multiple ribosomes. As soon as the protein is made, this kind of dissipates and you can't see it anymore. So we can look at the RNA and the protein together. And this is Roy, a graduate student in my lab, who did this. So here's the construct. We're putting these sun tags in there, and then we're imaging them. And so what we want to see is, here is the granule in blue. Then we have the RNA and the protein, and we can look if they co-localize. And this is an example. So this is what we're thinking, is that we have the protein being translated, so we make all these GFP constructs. But what I'm showing you here is we can actually show that we can bleach, so we can quench the fluorescence, so we can observe how RNA is translated, so we can get to the kinetics of how the translation is happening. And here you see this nice and slow. Now this was the bleach, and this is actually new translation. And so this means here's the ribosome coming and making these peptides, and we can get a lot of information about kinetics, and obviously we can use genetics also to figure out the mechanism of how this is happening. And what we can also do is we can look, because the RNA is long enough, if it was stretched, which we never really think that it is stretched, but if it's stretched, it's actually long enough that by imaging we can distinguish the 5' end, where the translation happens, from the 3' end, where the translation ends. And so a 5', when we look at a 5' probe, we see that that is mostly on the outside of the granule, so as the RNA is moving out. While if we're using a 3' probe, then the RNA, so here's the 3' probe, then it is mostly towards the inside. So what that tells us is that when the RNA is translated, it gets actually directionally exported out of the granule. And so then what Roy then did, he asked, what happens to RNAs which are not translated? And they're forming a circle. And this is really interesting, because for many years, scientists have been discussing that there are circles of RNA, and the question was, are they actually translationally active or are they translationally inactive? Our imaging, seeing is believing, that's at least my motto. Our imaging very much suggests that it is the inactive, non-translation RNA which makes the circles. Okay, so that was the kind of part where I wanted to tell you a little bit, because this is, I'm very excited about this work, as you can probably tell, but it's also because where the RNA is a tiny little part of the cell, and classical methods of biochemistry to address that question, you couldn't have used them because only 4% of the RNA is translated. 96% is not translated. That means if you squash up the embryo, you're going to test the 96%. You're never going to see what you're actually interested in. And so we had to kind of wait, because we've been working on this problem for a long time. We had to wait for the imaging technologies. And this is kind of, you guys are working on some of the newest technology. So often, technology allows you to do something. You have your problem, but the technology can drive you forward in your understanding. And so it's always really good to figure out what's going on technology-wise, and also sometimes to try to develop something, because you may not, with what you have, you may not be really able to show. So this is what I've been telling you so far. The germ cells, the germ plasm is pre-localized by the mother. Then the germ cells form. Then the germ cells migrate, and this is also, we've been studying the processes. This is really odd, and it happens pretty much in every organism, because you saw also in the mouse, the germ cells are forming sort of almost on the fringe of the embryo. And we speculate, we don't really, this is again evolution, you speculate. We speculate that it's because that keeps maybe the germ cells away from all the somatic signals. Because it is sort of an observation, the germ cells always form at some outside region, but then they cannot make egg and sperm unless they meet up with somatic cells, which make all the tissues which support the differentiation of egg and sperm. So they have to migrate. And then here they're meeting up. So here's the green are the germ cells, and there's all these somatic cells which give them actually the signal not to differentiate. Now you should differentiate, and that's hormonally controlled. And then finally in oogenesis in the fly, you have stem cells at the tip, and then you have these nice chains of developing egg chambers. And here you can see the germplasm already being formed. And so now I'm going to just switch to mouse and human development, because I want to tell you it's not that different. So we talked about at the beginning it's different, but otherwise there are many similarities with the germ cell. So what happens at the beginning? So here we have the zygote, which is of course the fertilization of the sperm with the egg cell. So now we have the zygote, and the zygote starts, and that's the very early embryo, just got maternal and paternal genome. And then divisions occur, rapid divisions occur, and then something is formed which is called the blastocyst, and this is a very, very early stage when actually the cells which are in this kind of cup here, they have not decided what they're going to be. This is actually the type of cells that many of you have heard when you make ES cells. That's where they're coming from. They're coming from that stage. And then development goes on, and we're focusing here on the germ cells. And again, the germ cells are forming, this is in the human, the germ cells are forming at this interposition between, squeezed into between these two tissues which are making mesoderm and epithelium. And then the germ cells also in the human, they have to migrate, and then they get to the gonad, and just like in the fly, in the gonad, they get surrounded by somatic tissues. What's actually important when they get to the gonad, surrounded by the somatic tissue, whether it's the fly, or it's the mouse, or it's the human, it is that the somatic tissue gives the cells the information whether it should develop into a sperm or an egg. So that's more important than the genetic information, whether it's an XX or an XY germ cells. So that's very, very important for the differentiation. And so here's again, this is the trophectoderm and the hypoblast. When you can, here you, at this, if you take them cells out of this stage, you can basically make stem cells which can give rise to every cell type, including germ cells. And this is the location of the germ cells, and you can see how it's squeezed in here on the side next to all these other cells that will make all these other tissues of the embryo, and much of this here is, again, the extraembryonic tissues. then the germ cells are migrating, so they have to actually get to this region. So they start off sort of at the end of the embryo again, and then they migrate into the gonad. And then in the gonad, dependent on whether it is an XY or an XX, they're going to start to develop very differently. So in the human or in the mouse, what is happening in the testis, they will develop into testis cords, which then make stem cells to produce sperm throughout the entire lifetime of the organism. While in the female, there are many divisions, and then the cells are arrested before they go through meiosis to produce mature eggs. So it's very different. In the fly, there's stem cell population for both. In humans and mice, there is no stem cell population. So all the eggs that are produced, or all the oocytes which are produced, are sitting there from early stages on. And we talked about meiosis. So this is also very different in the male and the female. In the male, this is a special division, and so one precursor cell makes actually 64 sperm, and while on the oocyte side, we also only get one ovum, and the rest of the cells actually often are thought to degenerate. And here is the final event, which is fertilization. And it's very interesting that until very recently, and it's still not absolutely sure, no one really knows how the sperm identifies the egg. So there was recently a sperm receptor identified, but more recent results show that that may not be the unique absolute thing. And so the probe for that was that if this is the receptor, and then and it has a specific interaction with the egg, and that's of course important that you only fertilize the egg of the same species. If you now clone that same receptor from another species, then if you express that sperm of this other species, it should not be fertilized. But when you switch receptor and ligand, then you should be able to fertilize. And that experiment is not completely reliably produced at this point. So we have a pretty good idea what may be the sperm receptor. And it's pretty amazing considering how important that question is. And a lot of people have worked on this. What's better known is how actually once the sperm enters, there's a reaction that then doesn't allow any of the other sperm to enter, and also how the sperm is attracted to the ovum. So just to summarize, so the primordial germ cells are set aside early during embryogenesis. Then they follow these attractive and repulsive guidance cues to get to the gonad. That's we're talking here about human, but that's the same for the fly. Then once they are in the ovary or testis, the precursors develop very differently. Meiosis mixes the genomes and establishes chromosome balance, obviously, between female XX or XY. And then oocyte attractants and sperm receptor control the excess, so only one sperm fertilizes an egg. And this is sort of more maybe a point for discussion. So many of you probably know that there are embryonic stem cell techniques. So either you can take cells from this epiblast that I was telling you, or you can even reprogram a fibroblast to become like one of these early cells in the embryo, and they can then make all kinds of cells. So they have been differentiated into neurons or muscle cells. More recently, people have also made organoids where you can make a little gut or a little brain, not very reproducible in terms of its exact tissue organization, but pretty good in terms of cell type composition. And so the question then is, can you make egg and sperm? And it turns out that now there are methods where you can make, certainly, and most of this is obviously done in the mouse, where you can take ES cells and make actually these primordial germ cells that I told you about. And basically what's really important is we know so much about the genetics of every step of how this process works that you can use these steps to make then these cells and then move them into either the sperm or the oocyte path. And there are now examples where this whole process has been made and where an artificially made sperm and an artificially made oocyte actually have been fertilized. This has to go in a culture. The oocytes are really difficult. This has to be done in a culture condition where there are actually some of these follicle cells around it that are normally there doing oogenesis. But nevertheless, this is at the end, the product, the fertilized oocyte is completely ES derived, and they can make an organism in very, very, very low percentage. So that means we don't understand everything. This is not like it's normally happening, but it can be done. And there was a report just this year where actually an oocyte was made from an XY, so which would be a cell which normally would make a sperm. And what they had to do for that is to lose actually the Y chromosome. Again, very, very low percentage, but this sort of is a possibility where you could say, you know, everybody can make an egg and a sperm, and I leave that for the discussion. So I just wanted to sort of come back to this evolutionary, that we have these commonalities and we have also some differences at the very beginning, which I was telling you about. And now I want to just make one more point, and that's sort of, again, the immortality. And kind of, I love this. I think it's the New Jersey Shore picture. So this tells us kind of genetics. You know, there's the paternal genome, maternal genome. You get half a copy of, you know, you basically get only one of the two copies of each of the chromosomal set that everyone, and then the little kid is a recombinant of that. And that kind of suggests that there is an equal contribution of father and mother. But if we think from a developmental point, that is not so. So eggs are gigantic in comparison to the sperm. The sperm mostly contributes the nucleus, while the egg, you can see how big, the yolk-filled, here, the chicken egg, they contribute a lot more. And so when we just look at volume, it's like an enormous difference in volume between the egg and the sperm. And the egg is really the original cell of the embryo. The sperm is one part of the DNA. And so we don't know how cells are actually made. We can make chromosomes now, can even fill them with chromatin, but we don't know how to make actually a cell. And so several hundred years ago, Virchow said all cells come from cells. We could actually say all cells come from an egg. And the egg actually gives much more to the early embryo than just the nucleus. And so in that cytoplasm, we have many cytoplasmic organelles, like ribosomes, which make the protein, Golgi, and ER, which transports these proteins to many locations, and cytoskeletal structures like microtubules, which help the cell to move or make an axon for the neurons. And then also, most importantly, mitochondria, the powerhouses of the cell, which have their own DNA, they're only transmitted through the egg. So there are basically no cases, no organisms where the mitochondria are transmitted from both parents. So and so without mitochondria, obviously the organism couldn't survive. So what the egg does, the mother does, is it provides actually all the information that you need for the early development of the embryo. And then the zygotic genome, the genome of the embryo, will start to make all those components, and they will be replaced. We know very little of how that happens. It's actually something we're really interested in my lab about. But the point is that we know so much about how the chromatin is organized, how the chromosomes are organized. We know much, much less about how the cytoplasm is organized. I just told you about these cytoplasm organelles. That's kind of like 10 years. that we kind of have been really grappling with. What's the role of these membraneless granules in the cytoplasm? And they're organizing gene activity. They're organizing what's happening in the cells. So, just a little, because I always feel like we can do so much in a culture dish. We understand so much about a lot of cells. Of course, when you really study lots of open questions. But I think germ cell biology, and that's kind of where there's the really positive part, and I think about recapitulating actually some of these events in a dish to understand much better what are the contributions and how actually the egg is specialized, how the specializations of the egg are produced. And with that, I have another sort of kind of take-home message, which is taking risks and making moves. So you heard, I mean, I actually was at MIT, then I went to NYU, and then back at MIT. And I think every time it was a great opportunity, and it's an opportunity because it is actually a good thing to change your environment. My lab actually started to do very different things when we were at NYU, and I'm sure now I'm actually very excited about a lot of the possibilities that I have at MIT. And so you should keep that in mind, even if you love the university where you are right now. And most universities have this kind of rule, right? As an undergrad, you have to go somewhere else. It's actually a good thing because moving, and those of us who come from a different country, the culture shock is kind of tough sometimes, but then it can also be very good because now you can actually operate into different cultures. So, and then what's also really important to me is community. And so when I started off, certainly there were, in my undergraduate class, there were certainly women. When I was starting to become a graduate student, there were many fewer. And when I became a professor, there were hardly any women. And so we have come a long way. We have a long way to go. But to me, building community, this is a celebration of my lab a couple of years ago. Building that community is really important to me, and sort of making a way of how everyone can get empowered to figure out what you actually want, what drives you, because I think it's actually pretty tough to be a scientist. There's lots of disappointments. Then from time to time you make a discovery that kind of what drives you. But that's not for everybody. And so I think it's very important to not say one particular career path is better than another. It is the career path for you, and you have to find out. And I think you probably, hopefully, are all provided with the opportunities to figure out what is kind of the career path which is the best for you, whether it is science, whether it's literature. There are so many aspects, and so many people in my lab actually have taught me how many different options there are. What can you do with a PhD? You could actually do a lot with a PhD, right? And so I hope you all find your way. And with that, that's my group now. And I thank you very much for listening, and I'm happy to answer questions. And I went way over time. So are there any questions, or was it too much, too specific? You can ask me anything. Yeah.
So I know sort of, I'm not a biology person.
Mm-hmm.
I want to know sort of what the implications of your research would be for sort of society at large, for future sort of, I guess what sort of impact are you hoping to have?
Yeah, so I mean, I think I am actually an example of a very basic scientist. And as I said at the beginning, germ cells are actually interesting, right? There are some germline cancers. They're certainly the pediatric ones, which are when germ cells get lost very early on. They're really easily treated. And then most of the cancers of the ovary, for example, or testes, are not the germinal cells. So in a way, what is the disease? So the disease that we're studying is really fertility and understanding the mechanism. And what I was telling you is kind of, they are unusual cells. And so I would say we really don't know much about really the biology of the cell. The other part that I'm feeling more where learning more, we making actually an impact is also because they're so different, right? I told you here is in an XY organism and an XX organism, you have a completely different developmental path. And that developmental path is also very, very different in aging. So aging oocytes, the cytoplasm doesn't work well. They can't separate their chromosomes. You get often aneuploidies, which, for example, leads to trisomy 21, which is Down syndrome. On the other hand, and other aneuploidies, in males, you get actually completely different defect. You get defects because the DNA, because of the replication, is affected. So I think the impact is really on a biological understanding, and the understanding and where this notion of how important aspects of outside of the nucleus are actually for reproduction. I think that is actually an important part. That's probably not what you. This is probably too scientifically what I. But that's kind of where I feel like where we make an impact. But it's always a tricky question because in a way, because the organism is not affected. It's the next generation that's affected. So it's sort of an interesting disease.
So you mentioned when switching labs that it was really the opportunities for people that brought you there and that. Can you elaborate a little bit more on what the difference in opportunities were?
When you go from one place to another? Yeah, so for example, your colleagues. Right? It's very easy because all of a sudden you have colleagues who do something else. So for example, when we went to, so there's two very concrete examples. So when we went to New York, I was surrounded by neurobiologists who were using actually imaging technologies that I had no idea about. I wanted to look at how those germ cells migrate, and we couldn't see it because they were in the middle of this yolk. And I would go to Germany and, Zeiss microscopes, they couldn't provide me. And so then I actually, through the colleague who was this he said, try my microscope, and it worked, right? And so that was and so I really got into much higher resolution, more depth, 2-photon microscopy. So that's one example. The other example is I had immunology colleagues, and we're also now working on an intercellular pathogen, and it was really by thinking about how, and it has also something to do with mitochondria, which used to be bacteria many long back. But how do actually evolve within the cytoplasm and other organism which can actually go from a pathogen to symbiont? I think I would have never thought about this if I didn't have listened to immunology talks and also sort of more people working on parasites. If I hadn't, so it's the juxtaposition, and it depends on what possibilities do you have. I think. Being at MIT, it's very exciting. I think especially we had quite a few discussions about AI. That is certainly a place where you can see you're going to have, I've already seen this. I have undergraduates in my lab who actually do a lot of segmentation and image analysis, and they're really excited about that, right? And those are the right people. So, I think it's just... I think that's the fun of being a biologist because there's just so much that we don't know, and hearing from people what they're studying, so you don't get into I have to do it this way, and also that helps actually your creativity. Some people can maybe be locked in the basement. I'm not one of those. I need, I like the stimulation. I like getting ideas, and often it's the transfer. It's actually the further you transfer the ideas, the more creative in a way, because nobody has thought about it, but you thought about it because you heard it in a completely different context and you drew it into your project. So, and I think that's probably irrespective. This probably applies to many other things than biology. So being open.
We'll take 2 more questions based on time.
Okay, let's go. Go ahead.
Thank you, Dr. Lehmann. Your talk was amazing. Big fan. So I study cancer biology. I don't know about germline biology. My question maybe is a little bit benign. But so about your work about RNAs in germ cells granules, are you investigating RNA condensations in these granules? Maybe that can be the reason why they are not translatable.
Yeah, so definitely we think that that has a lot of control, but it's actually cancer cells are, I mean, so that these granules are actually controlling. So there's a number of observations with granules. So certainly the granules can control whether you translate or you don't translate, right? How keeping RNAs through long times, which actually when you store the oocyte. But there's another aspect which is really interesting in granules. So having these cytoplasmic compartments could also change of how drugs are distributed within the cell. It may not be, the cytoplasm is not a gummy bear. It's much more structured than that. Gummy bear? So it's much more structured than that. What's really interesting, I thought what you were going to was like, I work on cancer cells, aren't they immortal, right? And what is very interesting is many cancer cells are actually expressing these RNA regulators of the germline. And if you mutate the RNA regulators of the germline, you can reverse that phenotype. So which is very interesting. So what is the connection to immortality is still a question. There were two more questions. Yeah.
Yeah, I kind of have a two-part question. I was wondering how exactly it's possible to take a cell from an XY individual and make it an oocyte, and also the applications of that. Like, can you take an XY embryo and turn it into XX? And then what are your thoughts on that?
I mean, so the way they do it is actually they use a repair mechanism that is faulty. And so it loses that Y chromosome and then duplicates the X. So you still have two X's because you need that to go through meiosis. So that's what's basically the trick. But yeah, I mean, the implication is anybody can have anything, right? So you can self-reproduce, which is, of course, not a good idea because then all those diseases which are... I mean, so it's the implications are more than we could think of in any science fiction movie, I think, right? So, yeah. So the problem with research is always a little bit, you love to do what you can do, but you do, right? And I think that's where, like, there's a lot of ethics. There was a lot of ethics discussions, obviously, about CRISPR, and certainly this is also something. I mean, which actually makes no sense from a biological point of view, because you wouldn't want to, you wouldn't want I'm going to make from myself some sperm and some egg, right? Which, of course, in a way, because the XX doesn't have a Y, right? So you can only do that from an XY cell. But, I mean, that's from a totally biological point of view. But you wouldn't want to do this because it's doomed to be really bad for the organism. And fortunately, since it's already so poor, I mean, I would say it's you probably can't do this. But in principle, you can. And this is sometimes you feel like, why did they do that? Right? But maybe it was the question, could you actually make an oocyte out of that cell? And as I told you early on, the PGCs, they're not that different, right? So I'm not so surprised about it. I feel like this is a typical, like, I can do this, and I'm